Hi everyone, this is going to be our final gene expression FRQ practice, and this one is going to be question number 4 from the 2016 exam. So let me pull that up for you. And this is a pretty straightforward question, I think. Maybe I should have done this one with you guys first, but save the best for last. So here we go. You have a picture here. The figure represents the process of expression of gene X in a eukaryotic cell. So eukaryotic means it has a nucleus, and you've got DNA showing introns and exons, and then going to a primary transcript, and then the mature mRNA, which is much smaller, gets out into the cytoplasm and turns into protein, or gets translated into protein. So part A of the question said the primary transcript in the figure is 15 kilobases, kbs long, but the mature mRNA is 7 kilobases in length. Describe the modification that most likely resulted in the 8 kilobase difference in length of the mature mRNA molecule. Identify in your response the location in the cell where the change occurs. All right, so in a eukaryotic cell, the mature mRNA is always going to be shorter than the gene. And why is that? And that is because eukaryotic genes have introns, and those introns need to get spliced out. So that's the modification. Now identify in your response the location in the cell where this change occurs. Intron splicing is one of the 3 events that has to happen before the mRNA can leave the nucleus. Do you remember what the other 2 are? One is intron splicing, one is a 3' poly A tail, and one is putting on that 5' cap. So those are the 3 things that have to happen in the nucleus before the mRNA can leave the cell. And part B, predict the length of the mature gene X mRNA if the full-length gene is introduced and expressed in prokaryotic cells. Now, prokaryotic cells, we're talking about bacteria. They don't have a nucleus. So we take gene X, this right here, and we stick it in a prokaryotic cell. Okay? What's going to be the length of the mRNA that the bacteria makes from that? Well, bacteria are incapable of intron splicing. So the mRNA is going to be as long as that primary transcript. It's going to be 15 kilobases long. It's going to be much longer. Justification is because prokaryotes can't splice out introns because they don't need to. Let's take a look at the scoring guideline. Describe the part A was worth 2 points. Describe the process. You could have said removal of introns or simply just RNA processing. Location, there's only one thing you could say for that. You had to know that that intron splicing out occurs in the nucleus. Part B was also worth 2 points, 1 point for the prediction. Now, you could have been very exact and say that the mRNA made in the prokaryote would be 15 kilobases long, or you could have just said it would be longer than the one made in the eukaryote. And the justification was that mRNA processing does not occur in prokaryotes. All right, so pretty simple, straightforward question. If you understand the mechanisms of transcription, I think you probably would have done pretty well in that question. Have a great day. Bye.